welcome back to A&P Live. Today I will be your host Harper, along with my colleague Adrian.
It's a great time in the sports world, Harper. Basketball season is back.
Yes it is, and LA has been known to have its rich basketball culture.
Of course, there's been some great players to come through LA, such as Kobe, Kareem, Shaq, and Magic Johnson.
So, did you know Magic Johnson had HIV?
Yeah, that's a really good point. It brings us into today's discussion about the immune system. However, there's a lot more to it. The immune system is only a small part of the entire lymphatic system. To start, we're going to break it into the 3 lines of defense.
Right, there's the first line, which has to do with non-specific defense and deals with like skin and mucus, so external and internal barriers. Or it even has, I think, white blood cells, which help defend as well, which are also known as neutrophils.
The second line of defense is also non-specific, and it uses a variety of cells such as natural killer cells, phagocytotic cells, antimicrobial proteins, and a ton of other cells to attack infectious agents that enter the body system.
So that would probably be like a response to a fever or like inflammation.
Oh yeah, when I sprained my ankle, my foot became really inflamed and red.
That's an inflammatory response, and that happens because there's more blood and lymph being pumped to the tissues, which is promoting healing and allowing for the affected area to be protected from an infection.
So what about when I have the flu? Is it the same?
Yes, but it's a little different. That is a second line of defense, but it's basically a pyrexia, which is also an inflammatory response, and it allows your body to naturally get over your flu. Then there's a third line of defense called specific immunity, which deals with when the body encounters foreign pathogens, which are also known as antigens.
Oh, so antigens trigger the production of antibodies, which secrete lymphocytes to clear out and fight off infectious agents in the body.
Yes, and now we're going to send you to our colleagues, which will have a further breakdown of these 2 defenses.
Alright, so to go into further depth and break down the specific defense, also known as the immune system, also known as the third line of defense, we're using an analogy of basketball and defense in basketball to describe it and hopefully be able to understand it a little bit better. So drawn here we have the half court of a basketball court, and we want to relate this to that third line of defense, like I said, also known as the immune system. So there are 2 types of defenses that you have within the immune system. You have humoral and cell-mediated, and in this case, humoral will be our perimeter defense, which is what's defending this outside area here, and cell-mediated will be the defense that's protecting the key. This is known as the key in basketball, or the paint, and your cell-mediated defense will be in here. We're referencing the key as, we're referencing the key as being inside of the cell, so keep that in mind. Humoral defense is known as your defense that's in the extracellular fluid, so it's outside of the cell, while cell-mediated is inside of the cell. So just keep that in mind, that the key is representing our cell. So what happens is, imagine that you have a pathogen that's like a basketball player trying to score a basket, okay? And you have a coach for the defense known as the helper T cell. In order for these 2 defenses that you have, both perimeter and inside the cell, they have to be stimulated by this helper T cell for the coach. So the coach has to give the orders so the players know what to do. Now, let's start with our humoral defense. So our humoral defense, like I said, is on the perimeter, and it involves B cells. There are 3 aspects of humoral defense. You have agglutination, neutralization, and precipitation. Okay? So with those 3 things in mind, we're going to apply that to this analogy right here. The first one is agglutination. Okay? So for those of you who aren't familiar with basketball, there's something called a half-court trap, where you can trap on defense, you can trap the offensive player within you can trap them within this corner, which is kind of known as a dead zone, and a place you don't want to get trapped in because you have nowhere to go. So agglutination will be like this half-court trap. Agglutination is when antibodies secreted by the B cells, or the plasma cells, excuse me, will latch on to the pathogen, in a sense, slow it down and mark it for destruction. So let's visualize this. You have the B cells, which differentiate into plasma cells and begin to secrete these antibodies. So these B cells that have been stimulated by the helper T cell recognize the pathogen, and they start secreting these antibodies that latch on to the pathogen. Okay. So now, in a sense, this is like a double or a triple team, where this pathogen now has a bunch of antibodies stuck to it, and it's, in a sense, trapped. But really, in your body, the cell can still move. However, it's a bit slowed down, and now it's being marked as foreign material in the cell. So for neutralization, which is the next step, this one can't really be visualized, but imagine dribbling a basketball, and then if you get stuck in this double team, most likely the attacker or the pathogen will pick up their dribble. In basketball, if you pick up your dribble, you can't move, right? You're, in a sense, neutralized because you don't really pose a threat, especially if you're this far away from the basket, okay? So, in a sense, neutralization here is forcing the pathogen to pick up their dribble and not really be a threat anymore. Now, the next step is precipitation, which in this case we're going to relate it to the basketball being stolen, which leads to the pathogen being stopped, and in the body that means that the pathogen would have been devoured by, phagocyzed by a macrophage and destroyed. In a sense, stopping the attack and preventing the pathogen from, in this case, scoring the basket.
So next we're going to talk about cell-mediated defense. Cell-mediated defense starts the same way as humoral or antibody-mediated defense. The helper T cell or the coach has to recognize where the pathogen is. Now, as we discussed before, in humoral defense, pathogen's out here in the extracellular fluid. However, in cell-mediated defense, pathogen has made it to this area or in the key within the cell. So, the coach has to recognize that and stimulate T cells that are located within the cells, or in our case, in the key. As soon as the coach recognizes that the pathogen's in there, these cytotoxic T cells are going to be able to attack the pathogen and prevent it from scoring or infecting the cell and reproducing. Now, in both humoral and cell-mediated defense, we have memory systems. So that's essentially the recognition of B and T cells of pathogens that they've seen before. So to relate it to our analogy, you might have basketball players that have played each other for a long time, and they know each other's moves, they know how each other work, right? So these T cells guarding a pathogen that they've seen before, or these defenders guarding a scorer that they've seen before, will be able to attack and effectively and efficiently stop the pathogens or basketball players from scoring. And it works with the B cells too. B cells are able to do that on the perimeter. The T cells are able to do that within the key.
And now, Starting at guard, measured somewhere between 9 and 12 nanometers, with a nucleus containing 3 to 5 lobes in an S-shaped array, the bacterial killer, neutrophil. The other guard, anywhere from 5 to 15 nanometers in length, with a nucleus that almost always leaves little room for cytoplasm, the T or B cell, the classic leukocyte, lymphocyte. At forward, measured somewhere between 8 and 10 nanometers, with granules for days, ready to secrete histamine and heparin at a moment's notice, the all about that base granulocyte, basophil. At forward, anywhere from 10 to 14 nanometers in length, with a nucleus that has 2 large lobes connected by a thin strand, ready to fight even the biggest and baddest of worms, the parasite destroyer, eosinophil. And finally, at center, making an entrance at nearly 15 nanometers in length, with a large kidney-shaped nucleus and abundant cytoplasm, still working to become a full-fledged macrophage, the giant, the pathogen eater, monocyte.